What? Which one? This one? So how do you change it to mine? It's already good. Can you go to my... Oh, it's switch on. Can I do it?
You're welcome to
Hello, welcome everyone. So this is a session on medicine and pharmacy sponsored by PAASE. So we have so far 28 participants, so that's good. So by the way, I'm Vince Faustino. I'm a pediatric intensivist at Yale. We have prepared for you an exciting panel of speakers and talks, primarily focusing on COVID. So we're going to split the session into two parts. So the first three talks are going to focus primarily on COVID, epidemiology, diagnostics, and therapeutics. We're going to follow that up, the three talks, with Q&A. So please post your questions in the Zoom group chat. And then for the second part of the session, we're going to have three talks about the how we're meeting the need for ventilators in the Philippines. So we all know that COVID is primarily respiratory disease. So when everything started, people both from the Philippines and the US started working as in groups to try to come up with alternatives for ventilators in the country. But before we go to the first talk, I just want to give, before I forget, we're having problems with YouTube. Well, I'm having problems setting up YouTube, so I apologize if it's not working. I'll try to fix it along the way. So hopefully it works, but if not, we're recording this and we're gonna post it later. So before we move on for the first talk, I'm just gonna show you a brief video on what PAASE is all about. I know most of you, our members, are familiar with it, but some may not. So this is a brief intro for everyone.
You have to go
Stop that.
share your.
Sorry about that.
It's on the road. Just move that one aside. And it starts really good.
Really. Stop.
Science and technology and innovation, these are indispensable in the modern economy, in the modern world.
We held a meeting at the MSI, and we resolved among about 6 to 8 of us, local members, first to form PAASE Philippines so that we could continue to host meetings here, but also to go on a massive campaign to try to get support from the national government through the legislative and executive branches to invest more in science and technology. So it started with a white paper, a position paper that we co-authored among 6 of us, namely Ernie Pernia, Cesar Saloma, Rador Asansa, Toby Dairita Fataneo, and Alvin Colaba de la Salle, and myself.
So PAASE historically, in the past 10, 20 years for sure, have been collaborating with our counterparts here in the Philippines at various universities. And a lot of these interactions probably started as peer-to-peer. But thankfully, we have the Department of Science and Technology here in the Philippines, and they've come up with some really helpful and very impactful programs that actually enable PAASE members that are based in the United States and other parts of the world to actually come here and do extensive research and academic partnerships and collaborations.
Well, I think that one of the good things that PAASE is doing is really trying to stay in touch with the researchers and the scientists here in the Philippines. So I think that is one of the good programs that they keep on supporting some of the initiatives of our researchers and scientists here in the country, trying to align also their assistance towards the plans of the Philippine government.
It's very important that the PAASE members and scientists based in the Philippines are continuously in contact, so we will know the needs of the Filipinos and opportunities that Filipinos can have to do science with their PAASE counterparts. So these interactions, such as the conference that we're holding and the annual conferences that we have.
The Philippines and the scientists there have very unique perspective that adds to discoveries and inventions, advancing in general the field.
These members, because they are part of an academy, it implies that they are productive. So they have publications, they have innovations, they have inventions, patents, and they have, they have an impact on society. Okay, so these are the people who can convince government, invest more in science and technology. And that was really the gist of our paper, our position paper in 2000. And it is Ernie Pernia who said that R&D by nature is expensive and it is a public good. So it is important for the government to take the lead in investing in R&D.
To increase the economic growth potential of the country, we cannot continue to just rely on traditional inputs into production as capital and labor. We also need to develop our expertise in science and technology and innovation so that we can come up with new systems, new ways of doing things.
I think that science, researchers, industry, and even the government is really like an ecosystem of knowledge. So I think it doesn't really matter if you're coming from a private institution or a public institution, such as a state university. I think we all have a role to play to improve the research capabilities and scientific knowledge here in the country. I think it is about time for us to realize that one of our roles as academics, as researchers, as scientists, is to actually contribute to that global body of knowledge.
And the role of this cooperation among these government agencies really is to be able to attract science and technology, global-based companies to set up shop here. And that is really key in terms of developing the science and technology innovation ecosystem in the country.
Particularly because there's that deep desire. And what PAASE does and can do more of is to promote this relationship between its members.
We really need to ratchet up the appreciation, the recognition of the importance of science and technology and innovation.
And we are all trying to attain what we call the common good, the greater, higher, long-term good of the Filipino people.
Okay, so that's a quick overview of PAASE. So now to start the session, we have Edsel Salvana. So Edsel is one of the top infectious disease specialists in the Philippines. He's going to give us a brief overview of the current epidemiology of COVID-19 in the country. Edsel, the floor is yours.
Okay. Good morning, everyone. Thank you to PAASE for the opportunity to present an overview of what's going on in the Philippines. And as you know, this has been going on for a while now, since January. And this is just really kind of a snapshot of what's going on, and a little bit about what's going on in terms of transmission and some of the genomic work that's happening at the Philippine Genome Center in RITM. So just very briefly, our first cases in the Philippines started towards the end of January, and these were all imported cases from China. At that time, we eventually shut down borders with China, and these patients, it did not look like there was forward community-based transmission, because there was a lot of surveillance at that time, and it took about more than two incubation periods before the next cases came in. So epidemiologically speaking, those first three cases were distinct, at least from those first three cases in January. The first death outside China, unfortunately, did occur in the Philippines, and this was one of the three Chinese nationals. He also had influenza and strep pneumo on top of the COVID, and we think that that's why he died, aside from some of his comorbids. In March, the start of March, we had two new cases. One was imported from Japan, and the other one was the most concerning one, because he did not have any travel history or a known contact with a positive case. And at about 10 cases, this is when some of us on the technical advisory group of the IATF already urged the government that if we found one that looked like it was community transmission, we should think about closing down. At about 52 cases, we actually declared GCQ and DCQ right after that. In early March, we peaked at 538 cases, and then the cases started slowing down, although there was a lot of delay in testing as well. And then as the cases started to decrease on daily cases, and there was an indication of a slowing down, then ECQ and GCQ were modified. In June, we saw a second epicenter occur in Cebu. There was also more expanded testing, so the cases were also going up due to expanded testing, but even correcting for that, there was really an increase in cases. And in July, the cases continued to increase. The Cebu cases have actually started to decline. That's why they were able to go down. NCR continues to rise, and the concern really is that our healthcare capacity is approaching critical levels. Available meds right now, just as a snapshot, we do have remdesivir, tocilizumab, favipiravir, dexamethasone, high-flow cannulas. We're no longer using hydroxychloroquine or lopinavir, ritonavir, just in case that comes up later. So from yesterday's number, we have 85,486 cases in total. 90% of these are mild. We have about 56,000 active cases right now. Every 15th and 30th of the month, the DOH actually cleans up some of these cases as recoveries if they've been on the board for more than 4 weeks and we knew that they were mild. A lot of those automatically get assigned to recoveries. So there's probably going to be more recoveries later, is what it's going to look like. Again, the cases last week, they were breaching 2,000 almost every day, but now we've had 1,600, 1,800, and now hopefully we start to see a slowdown on these. Again, a lot of these are delayed as well because it comes from all over the country. And on top of that, you also have some of the labs reporting late. And you can see the recoveries that are written there as well. Now, NCR capacity is getting worse, and we can see this on the ground for those of us who are in hospitals, and even the ERs are starting to fill up. So we really need to increase our capacity at this point. And if you smoothen out the numbers, because some of these are delayed, this is confirmed COVID cases by date of illness. You can barely see the January 16 to 31 over there, but the rest, you can see there was a real increase. From March to June to about end of May, you can see the NCR numbers, which are green, were fairly flat, and then started to rise up again. But the initial increase in cases in May was really driven by cases from Cebu. Just a weekly snapshot of it, and you can see again that right now NCR seems to be driving a lot of the cases, and the blue region, which is mostly driven by Cebu, is starting to go down. And also, if we look at it by region, you can see that NCR still has the bulk of the cases. Visayas had that blip, but again, it's starting to go down. And then Mindanao has actually had a slow rise. And just looking at cases by sex and age, you can see that most of the cases occurred between 20 years old and about 50 years old. Now, some of those may be artifacts as well, because if children and younger people don't have very severe disease, or if they're asymptomatic, they're less likely to be tested. But you can also see that the ones who died were in the upper age groups more than the lower age groups. And if you look at the graph at the bottom, you can see that the deaths actually have a bimodal distribution. The deaths peaked early on in March and then came down, and then peaked again, and now it's going down again. But with this increase in healthcare capacity, I'm pretty sure it's going to go back up again, unfortunately. And just case fatality rates, you can see this is consistent with what's in literature. Above 70 years old is really kind of a horrific number of a percentage of deaths. They do not do well, and it's just like SARS when we're talking about older age groups infected with COVID-19 in the Philippines. And testing has actually improved. Even the backlogs are getting less. Where they've tested more than 1.3 million samples, but understanding that these tests are not perfect. PCR using nasopharyngeal swabs is about 62% sensitive on the day of onset of symptoms, and is about 80% on the third day of symptoms, and it just goes down after that. So we may not be capturing everyone. You can see that the positivity rate was pretty high at the start of the epidemic, and this was because we had very little testing available and we were prioritizing symptomatic patients. It went down and then has slowly gone up again, and it's about 9% at this time. Just a little bit about what's going on in terms of the genomes. This is from NextStrain, and I talked to Dr. Saloma, and she said that this is not complete that's in NextStrain because they have an algorithm that actually samples what's in GISAID. So what's in NextStrain here is about 10 whole genome samples from the Philippines, and you can see that there were about 8 samples around March, and to the right is the samples that came between June and July. And you can see that there was some clustering in the samples between March and April, but there's actually that kind of an outlier on top over there with the 20B clade. That's more of a European clade, and we'll talk a little bit about that since I still have about 6 minutes. So just looking at that 20B clade that was sequenced sometime in June, I'm not 100% sure if it was collected in June, but that's when they sequenced it. And so the provenance of this one actually seems to come from Germany. So this is the first European origin whole genome sequence that they reported in June. If we look at the others, there was that clustering early in March, and then there's another one above that you can see. And then there are two sequences that seem to be related, one from March and one from June. And this might actually is probably a community transmission that that continued in the Philippines because they're so close together. But you can see that there are different clusters that have been seen in the Philippines, and this is consistent because we have a lot of OFWs coming in on top of local community transmission. So there, aside from the first one in January, and then the Japanese and the local transmission, wherever that came from, there have been subsequent waves of establishment of these viruses, particularly because we've had more than 80,000 OFWs come in. The early ones, including the second sequence from January, from June, actually seemed to have an Indian origin, and of course this came from China. Again, with the algorithms of NextStrain, there are some limitations because they switch out some samples because there's a maximum set number of samples per country. And so in the past, this is actually also clustered to Japan, then India, or Australia, then India. But right now, the clearest one is China, India, Philippines. And again, this might be just a bioinformatics artifact more than anything. But these are the closest related samples to that, to those 9 other Philippine samples that are in NextStrain, aside from the one that looks like it came from Germany. Now, there's an interesting paper that came out that I think that is actually very controversial, and this is the shift of the mutation D614G, which is on the spike protein that supposedly makes SARS-CoV-2 variant more infectious. And there's a couple of reasons they thought about this. Is first that they saw a global transition in most of the sequences from D614 to G614 to the point that about 95% of the samples that they're sequencing all over the world in June to July is all G614. The second is that they've actually seen lower RT-PCR CTs for G614, which translates to higher viral loads in patients. And this looks like it might be a fitness advantage. There's no evidence that it's deadlier, but it may actually be more infectious. And they actually did pseudotype virions, and the virologists in the group can explain this better. But my understanding is that they basically mixed the G614 spike protein onto a different kind of virus, and they used it for experimental infections in mice, and they found the pseudotype virions with G614 variant were more infectious and grew to higher titers. This is because G614 seems to stabilize the spike protein. The D614 seems to break off easily, and G614 seems to stabilize that so that it gains entry into the cell easier. So you can see that if you look at the G variants, there is one that is found in NextStrain in the Philippines, and that's the one that came from Germany. All the other ones were D variants. But again, this is 10 sequences. And in June, one sequence was D and the other sequence was G. And my understanding is Dr. Saloma and the PGC group have actually seen a lot of G variants as well in the newer samples using partial genome sequence for the spike protein. Now, just this is going to be a quick video that will show you how the blue, which is D, becomes eventually replaced by G. So watch out for the brown to start coming in. This is about a minute. You can see above there's like the timeline, January going to February. And then you can start to see that some of the brown stuff is showing up in North America as well as Europe, and also starting to even get into Australia and even Malaysia. And now you're seeing South America turning brown. And this is, as we go on in March, it's almost completely replacing the blue-green D variant and is turning it into the G variant. And as we go further, you can see almost all of the infections, even ones in China, are now G variant because they're being imported back into China after it mutated. Now you can see that there's almost, and now Philippines has half because in June we only had 2 sequences in next strain, and one was a D variant and one was a G variant. So this goes through, and now you see almost everything is G. So it's very concerning. And if this is indeed more infectious, as has been suggested by the Cell paper, then we're really in a lot of trouble. So in summary, the Philippines has had an initial cluster of cases in January, followed by sustained community transmission in March. And there was a major surge in cases that was mitigated by ECQ. The estimates of the World Bank are that we were able to avoid 1 to 3 million cases and about 100,000 deaths. Unfortunately, this had severe economic consequences. The case fatality rate has been improving. It's about 2% right now. We hope that it's going to keep going down. The cases, unfortunately, are surging anew as we open up. And there's a second epicenter in Cebu, but most cases are still in the NCR. The G614 may be more infectious, so we're getting hit with a double whammy. We're opening up, and then we have the G614 here in the Philippines. And so, it doesn't spell good news for our healthcare system, and we have to do better with other things that we can do that don't have such a major impact on the economy. We really have to maximize our prevention measures, rationalize testing, increase tracing and isolation efficiency. We need a vaccine, but in the meantime, actually treatment is getting better. And the goal is to preserve the healthcare system and keep the deaths low at this time. Thank you very much.
So thank you very much for that very interesting talk. I just would like to remind everyone, please post your questions in the Zoom group chat. We'll have a question and answer portion led by Maima Albaida, my co-host, after the first 3 talks. So I'm already seeing a couple of questions come up. Please continue typing them and we'll address them after the first 3 talks. So our next speaker is Charles de la Cruz. So Charles is pulmonologist / intensivist here at Yale. He's part of the team that developed the SARS-CoV-2 diagnostic testing test using a saliva. So Charles, the floor is yours. Okay, Charles,
can you hear me?
Yeah, good. Okay.
Yeah, thank you very much for inviting me to present to the PAASE group, and congratulations on your anniversary. I was asked to talk a little bit about saliva, specifically on diagnostic testing for SARS-CoV-2. You just heard in a nice talk by Dr. Salvana how Philippines is doing with regards to cases and death. This is updated this week. Unfortunately, in the United States, our numbers are incredibly high, and then globally, we all know what's going on with this pandemic. Very early in the pandemic, early January, the sequence for SARS-CoV-2 was identified and listed here. And some of these are the viral genes, the N gene, the E gene, the spike gene for the receptor bindings is quite important, have been sequenced. This plays into a role importantly because of diagnostics. And in terms of diagnostic currently for COVID-19, we can think of it at the different phases of the disease, from screening asymptomatic patients to monitoring contacts, to identifying patients who are symptomatic when they come into the hospital for diagnosis, and then later on for whether or not, how do we de-isolate and monitor for shedding in the convalescent phase. Diagnostic testing can also come in many different flavors with regards to, for example, antibody testing in the blood for IgM, IgG antibodies using the rapid tests or ELISA testing. But that won't be the major focus of this talk. I'm going to be talking specifically more about PCR testing using nasopharyngeal swabs, and then how that compares with saliva. in the detection of viral RNA. And this has become sort of the gold standard in diagnostic testing because it sort of detects, it's rapid, it detects the viral presence. I mean, in the olden days, I think you would do viral cultures to see that you actually have live viruses. But given the need for diagnostic testing, these are more sort of molecular these days. These tend to be labor intensive. There's a lot of reagents, specialized equipment required, and then potentially sampling error. Specifically, for example, for molecular testing, you would require the specimen collection, RNA extraction, deactivating the virus, finding a place where you can do this safely. And then you have the results of the test based on cycle threshold, as you can see on the bottom right. And the lower the number, the higher likelihood of the viral load in your patients. And then the 40 cycle threshold typically are the cutoff to be anything less than 40. Most labs find it to be positive. And then the lower the number, the higher the viral load. And you can see in the middle bottom here, for example, in this one, they use E genes. The N gene is used in many of the diagnostic testing. Other genes like the RNA-dependent RNA polymerase gene are used for confirmatory. And others are also using, for example, RNase-P, for example, as a gene to measure hosts, the patient sample as a control. And then you have a machine like this, for example, to do the PCR. In the detection of respiratory pathogen, for example, you have the nasal pharyngeal swab. I don't know if you have had it done, and most of the time it's quite uncomfortable and irritating, and it's been the gold standard currently. Other people have tried oral pharyngeal swab, which tends to be more tolerable. It still has its irritation, and at least in the CDC, the recommendation currently has been still the use of nasopharyngeal swab, and then there's some thoughts about using other samples, for example, sputum, endotracheal aspirates, BAL samples, for example, of patients who are intubated. So what's the connection between positivity of PCR and viral culture? And then very briefly, in the upper left, you can see that the higher the copy numbers, the more likely you're going to get positive cultures. In addition, in the bottom here, the days after onset of symptoms, as you heard earlier, the less likelihood you can get positivity on the RNA, and then this goes along with the positivity on the positive viral culture. In this publication here, you can see these positive culture in open circle tends to have lower cycle threshold, which goes along with higher viral load, more likely in a PCR to get positive culture. And what's interesting is that the pre-symptomatic patients, at least in this small number here, they have low cycle threshold, but then also you get to have positivity, which sort of highlights the importance of these pre-symptomatic patients to be highly contagious and transmittable of their disease. And that's why social distancing and masks are really important in the control of this virus. So then why is saliva important? It's you can collect all the oral fluids. It's been done in other detecting other viruses, for example, including coronavirus, with fairly good concordance. It's quite efficient in screening. People can use it in the absence of airborne isolation, for example, and healthcare workers are not necessarily required. You can ask the patient to do this. It's very simple to do. Eliminates the wait time for the specimen collection, and it's actually quite good for serial viral load monitoring because it's less uncomfortable. And so I think it's a great idea to explore the possibility of using saliva. Other studies have shown that, for example, you could detect saliva in self-collection in this small number of patients, and you can see the numbers here over time of hospitalization sort of tend towards a decrease in numbers. You have one patient here that actually goes to 0 and goes up again, and those tend to be, sometimes you're concerned about sort of false negative, right? And so I think you try to look for those kind of patterns, and you can see positivity in most of these patients. So at Yale, in the beginning, and as everybody else, we have collected positive COVID patients starting in March, both in hospitalized patients who are tested positive, recruited a lot of their samples, including sputum and saliva, along with blood samples. Today, I'm going to be focusing mostly on the saliva. In parallel, we were collecting samples for patients, for healthcare workers who are working in our ICU and sort of COVID floors as well, with high occupational risk, and then screen them for transmission. And so these healthcare workers have self-administered NP swabs, as well as sampling every 3 days. Typically, these patients are asked to spit into a sterile cup upon waking up. Some of the advice have been to not to eat, smoke, or do oral hygienes before 60 minutes before the collection. And so some of the early studies suggested that there's actually pretty good matching of NP and saliva, and the positivity of these patients are around 60 to 77%. These include all comers, right? As patients come into the hospital, they're all positive, and over time, their positivity decreases. And so, but you can see the concordance is actually quite good. This work by some of our colleagues, and Wiley, is currently in medRxiv as a preprint. And what they saw they saw quite strong SARS-CoV-2 detection in saliva here in the left. You can see the copy numbers tend to be a little higher than nasopharyngeal swab. And when they match these samples, again, they tend to be higher in the nasopharyngeal swab compared to NP in the saliva. And what's interesting is the false negative rate for NP swab was actually quite high. It's 124 in the matched samples, whereas the false negative rate for saliva is only 12%. Suggesting that maybe you can get the same positivity with a less negativity rate for saliva if you use it for testing. And so this is currently under review. In the same study, they showed that the saliva, where in the saliva cohort, you have negative tests. You can see on this one sort of line here, where one time is negative and the next day, next several days is positive again. You see more of that in nasopharyngeal swab. And it's likely possibly technique that especially, and these are included subjects who are self-administered a nasopharyngeal swab, and so the reliability of the saliva is actually much higher. And then you can see here in the healthcare worker and the left-hand side, the saliva SARS-CoV-2 copy numbers are actually, you can see the spread. And what's interesting is that in the healthcare worker self-sampling, the variance in the saliva sample is actually much better than the nasopharyngeal. So there's bigger variance here, where you can see there's a lot of differences between the different time points in the healthcare workers. And others have shown that the NP variability is actually not quite present in other studies. This is a study from China where they look at nasal swab versus throat swabs and see that even throat swab is actually a lot worse. The detection of CT values are actually a lot higher, and they're not able to detect it where they can see in nasal swab. They didn't do any studies in saliva. There's some early meta-analysis of saliva PCR testing, and I listed some of these publications here. In general, the sensitivity that they've reported ranges from 78% to 100%. The specificity, the only thing that they can say is that there's one positive saliva that in 50 negative NP swabs. In another study, was 2 positive saliva and 98, suggesting that the specificity is actually quite good. Others have found sort of higher viral loads in NP swabs versus saliva. And so I think there's more studies that need to be done to really find out the use of saliva. Some of the limitations have been most of the studies are not focused on early in the disease course, and the performance in the outpatient settings. There's a lot of work currently, even including Yale, about asymptomatic individuals and early illness and whether the use of saliva could be very helpful. There's comparable study in the outpatient that looked at saliva as a diagnostic. The Rutgers University has a expanded use already on 60 patients, and then there's other places that are starting to use saliva. If you look at clinical.gov, there's almost like 50 ongoing trials using saliva as diagnostic testing. Here I just want to highlight the use of NP swabs, for example, looking at viral load. And one of the messages that is coming out is that the viral load is actually much higher in patients with severe disease in red here. You can see even 13 days after onset of disease compared to mild symptoms. our own study, and this is led by one of our immunologists, Dr. Iwasaki, has shown similarly that the severe cases in our ICU has more persistent higher viral load. They're working now on looking at the saliva and to see if that could be done. So where to go next? I think the COVID outbreak is continuing, unfortunately. Hopefully we have a better handle of this. It's disproportionately impacting vulnerable societies. You know, I think saliva potentially has an inexpensive, simple way to scale the testing that's being made available in other countries. And so there's a lot of exploration on this area. It still requires some development. There's still some risk of handling saliva samples because of the viral load. There's a question about stability of the RNA in the saliva, and I'll be talking about antibody detection. A lot of people are now working on the validation of point of care and home testing for diagnostic platforms. One of the things that we found is that the saliva could be seen, if it's fresh, you freeze thaw it in 30 degrees Celsius or room temperature, you can see the stability is actually quite good, which bodes well for its use in diagnostics. Some of the things that also have been developed is something called the loop-mediated isothermal amplification, which actually is kind of interesting because it can detect a very low viral RNA in a sample, less than 10 genome per microliter. Yes, so this has been sort of in a preprint showing this handy fuge lamp to detect saliva virus. It doesn't require specialized equipment. They have a way of making this centrifuge. You can see this Eppendorf tube that can spin, and you don't really need electricity. You can get a rapid readout in 1 hour. It's really quite cheap and $1. And it's a pH-based colorimetric assay where you could see the colors change from the pink to the more orangey yellow color in terms of copy numbers. And so these kind of technologies actually would allow for more rapid testing and in a very cost-effective way. On the right and the bottom here, people are using a different technology, using nanoparticles, where they could see these aggregates form when there's positive viral RNA. And then when these aggregates form, there's a color change. So I think these are kind of pretty cool and exciting ways of doing these diagnostic testing in the saliva. At Yale, they're working on sort of more streamlining of this to try to multiplex with PCR. Some are also already doing it in conjunction with influenza to detect different viruses, and then to validate some of these in sort of asymptomatic patients. Some of the considerations are, the diagnostic needs versus research. For example, is this sufficient for detecting asymptomatic infection? How do you collect these in specialized tubings? And I'll show you some example. And then some of these strategies in terms of testing, can you pull these testing so that you can have sort of a small community pull the testing, and if there's a positive, you can do more fine tuning rather than doing individual testing. So people are trying to strategize how to make it efficient so that you don't have to do a lot of testing if you want to know what's going on in your community. I know at Yale, they're testing sewage samples in our community to find out any positivity, to get a sense in the sewage material, whether there's any. A couple more slides. These are some examples of how to collect the saliva. There's different sort of companies that do it. The Omnigenes are one of them that we've been using for one of our clinical trials. In fact, some people say that even the regular sputum cup would be enough because you don't need some of these. People are working on things that make it less complicated. And so other things to measure, I talked about the virus. People are thinking about measuring antibodies, maybe biomarkers like proteins. Just a quick, I don't have to go through into details. There are already some studies to suggest you can actually measure these anti-spike IgA, IgM, IgG antibodies in saliva. As you can see on the right, people are now correlating serum and saliva. And so as a diagnostic test, just to give you some flavor of what's going on. So in summary, I think saliva-based diagnostic is quite a promising alternative, quite non-invasive, easy to collect. Potentially economical. I think what's important is that this could be used as in home use, self-collecting, multiple sampling, and really possible real-time diagnostic, especially with the LAMP technology. And so thank you very much. I'd like to thank some of the investigators who did a lot of the work on the saliva testing, and some of the collaborators with this Yale Impact Team, which was formed at Yale to really counter the coronavirus pandemic locally. And thank you for your time.
Charles, thank you very much for that very interesting talk. I remember like early on, I was seeing some of the email exchanges, the questions about saliva, and the first article I saw was from your group, so it's very enlightening. So for those who just joined in, please post your questions in the group chat. We're gonna have a Q&A session after this next talk. And again, I apologize for not being able to live stream this through YouTube. We're having technical difficulties, and I don't know whether we can fix it on time. So anyway, so our last speaker for this for the first half of our session is Maricar Malinis, a very fitting name for an infectious disease specialist. Maricar, I believe, heads the COVID treatment team at Yale. So she's going to update us on the evolving treatment approaches to COVID-19.
Thank you, Vince. Good morning, and I want to thank Vince Faustino and the organizers for giving me the opportunity to give this talk. So I will be discussing the evolving treatment approach to COVID-19, and day to day, there's always new data. So there's, even this morning, I have to make some updates to my slides. So I have no disclosures. So before I proceed with the discussion of therapeutic management, I will discuss briefly the clinical presentation of COVID-19, which has been slowly understood with time and clinical experience. It is theorized that the disease exists in two phases. The first phase is the viral response phase, and the second phase is the host inflammatory response phase, or otherwise called cytokine release storm, as depicted in this illustration. How does this translate to clinical presentation? So during viral replication, infected individuals may have mild constitutional flu-like symptoms with or without GI symptoms, and lab abnormalities may include lymphopenia, increased D-dimer, and LDH. The inflammatory response may manifest early with pulmonary decompensation with evidence of abnormal chest imaging, and also may present with liver enzyme abnormalities. Worsening inflammatory response is likely when there is pulmonary and extrapulmonary manifestation, including ARDS, systemic inflammatory response syndrome, shock, and cardiac failure, with notable elevated inflammatory markers that may include CRP, LDH, IL-6, D-dimer, and ferritin. Awareness of this pathophysiology and clinical staging of COVID-19 can assist in the clinical decision-making with regards to choice of therapeutics or intervention. So antiviral therapy is preferred to be given early in the disease course, while immunomodulatory agents have a role once the patient hits the inflammatory response. At present, there's no US FDA-approved treatment for COVID-19, and supportive care is considered the standard of care. So in response to the pandemic, the Yale School of Medicine and Yale New Haven Health Multidisciplinary Treatment Group produced a standardized treatment approach to the management of hospitalized COVID-19 patients. In this treatment algorithm, which has been frequently revised based on the most up-to-date clinical data, local clinical experience, and expert opinion by a multidisciplinary group composed of specialists in infectious diseases, pulmonary and critical care, rheumatology, immunology, and hematology, and of course, our reliable pharmacist. This treatment algorithm, as mentioned, is specific for hospitalized patients with confirmed SARS-CoV-2 by PCR and requiring oxygen supplementation. So patients who meet this criteria will receive both remdesivir and dexamethasone, or its steroid equivalent, and tocilizumab has been added as a rescue drug. Rationale for this use will be discussed later in the talk. So in addition to the treatment recommendations, We also recommend routine lab monitoring and EKG to assess renal, hematologic, and cardiac complications, respectively. Of note, we have also given recommendations of anticoagulation and aspirin because of the observed increased thrombotic complications in these patients. There have been several agents that have been suggested to have in vitro activity against SARS-CoV-2 based on either prior data with MERS and SARS-CoV-1 and through computational science. Of note, atazanavir, a protease inhibitor that was previously included in our algorithm, was removed once the New England Journal of Medicine study revealed that lopinavir-ritonavir, which is a protease inhibitor 2, was shown to have no difference in the clinical improvement or mortality when compared to standard of care. Some of the mentioned drugs here have limited clinical data to assess their efficacy. Camustat and favipiravir are currently being evaluated in clinical trials. Today, I will discuss the therapies that have clinical data that defines their role in the management of SARS-CoV-2. So first, I will address the most controversial drug, which is hydroxychloroquine and chloroquine. So chloroquine is a known anti-malarial drug that has in vitro antiviral effect against novel coronavirus that inhibits viral entry via ACE2 receptor and prevents acidification of endosomes, therefore interrupting cellular function and viral replication. In addition to the antiviral effect, it was speculated that it may have an immunomodulatory benefit by attenuating the cytokine production and inhibiting the lysosomal activity in host cells. Early small studies during the pandemic supported the use of hydroxychloroquine by demonstrating higher rate of viral eradication, shorter time to clinical recovery, and radiographic improvement. However, more recent randomized controlled trials have shown no clinical benefit of hydroxychloroquine when compared to standard of care. The recovery trial in the UK have shown no 28-day mortality benefit when compared to standard of care. The ORCID trial that was sponsored by the US NIH was stopped once preliminary analysis revealed no benefit of hydroxychloroquine compared to placebo. And a recent study also showed no benefit of hydroxychloroquine in mild cases treated in the outpatient setting. Another concern with hydroxychloroquine is the QTc prolongation and possibly cardiac arrhythmia when used in combination with azithromycin, as demonstrated by the study of Borba in Brazil. The US FDA, who initially issued an emergency use authorization for hydroxychloroquine, has to withdraw this authorization. And the NIH and the Infectious Disease Society of America guidelines do not recommend its use outside of clinical trials. One of the promising therapies from remdesivir, which is a nucleotide analog that binds to viral RNA-dependent RNA polymerase and inhibits viral replication. It was initially developed in response to the Ebola outbreak in West Africa and has in vitro and in vivo activity against MERS and SARS-CoV-1 and in vitro activity against SARS-CoV-2. The ACT-1 study, which is an NIH-sponsored study, involved 60 sites, published the results recently in New England Journal of Medicine. In this study, 89% enrolled were considered to have severe disease, which meant requiring some form of supplemental oxygen. There was shorter recovery time overall in those patients who received remdesivir compared to placebo. When further analyzed based on baseline ordinal scale of clinical severity, those who were on supplemental oxygen had the greatest benefit from remdesivir. This study showed some trends towards improved 14-day survival in remdesivir arm, however, it did not reach statistical significance. Based on the result of the ACT study, the US FDA issued an emergency use authorization for remdesivir, and the US federal government allocates remdesivir to the states based on the number of active COVID-19 cases. The immune dysregulation and the exuberant immune response in SARS-CoV-2 has been shown to cause lung damage and overall poor outcomes. Target immunomodulation can ameliorate the cytokine storm, pulmonary inflammation, and may improve overall survival rate. There are several drugs that were speculated that can attenuate the hyperinflammatory phase of COVID-19. Currently, the NIH and the Infectious Disease Society of America guidelines have emphasized that there is lack of clinical data to recommend these listed agents, and it's preferable that these agents are offered in a clinical trial study. More recently, the sarilumab clinical trial in the United States, sponsored by Sanofi and Regeneron, was stopped after the drug failed to prevent deaths or get patients off ventilation, which were the study's key endpoints. More so, patients treated with sarilumab was found to have severe side effects, including shock and multi-organ dysfunction in 3% of enrolled subjects. Now, IL-6 is a cytokine secreted by monocyte and macrophage that appears to be the main driver of immunologic response and symptoms in CRS based on early case series of COVID-19 in China. Now, tocilizumab is a humanized monoclonal antibody similar to sarilumab that inhibits membrane-bound insoluble IL-6 receptor and has been approved for rheumatoid arthritis and cytokine release syndrome related to chimeric antigen receptor T-cell therapy. A small study from China reported that 21 patients who received tocilizumab had fever resolution, decreased oxygen requirement, improved chest CT imaging, and inflammatory marker profile. In our institution, we included tocilizumab in the treatment guideline because of some clinical data and its safety profile based on the experience of our local hematologists who have given tocilizumab for cytokine release syndrome after CAR T-cell. The data collected was during the first 3 weeks of the COVID surge. We evaluated survival and mechanical ventilation rates at 14 days. During this course, we have shifted from giving tocilizumab in critically ill patients to giving it early when there is increasing oxygen requirement with rising C-reactive protein. Note that the graph on the right illustrates our mechanical ventilation rates that have been stable despite increasing number of COVID cases. The survival of patients with severe disease who received tocilizumab was similar to the non-severe cases. As one of my colleagues have said, the tocilizumab seemed to be the equalizer. Randomized controlled trials of tocilizumab are still ongoing, but this morning, preliminary results from the COVACTA trial, which was sponsored by Roche, was released to the press. The trial was a randomized controlled study of tocilizumab versus placebo in the treatment of severe COVID-19. The primary endpoint of the study was 28-day improvement in clinical status, which was not shown to be any different in patients who received tocilizumab compared to placebo. There was no survival benefit nor difference in the ventilator-free days of those who received tocilizumab compared to placebo. Although notably, the time to hospital discharge was shorter in the tocilizumab arm compared to placebo, but the authors commented that this is considered not significant since this was not, the study did not meet their primary endpoint. Final paper is still in the works and still needs to be peer reviewed. Now, steroids have been used in respiratory viral infection and has been controversial in general. The use of steroids in influenza had been associated with increased mortality. In the MERS and SARS-CoV-1 experience, there was a decrease in delayed clearance of viral RNA from respiratory tract and blood, respectively. In contrast to other novel coronavirus, there was mortality benefit observed in the use of methylprednisolone amongst patients who were mechanically ventilated with ARDS per study in China. The recovery trial was designed to evaluate the effects of potential treatments in patients hospitalized with COVID-19 in the UK. And this was supported by their National Institute for Health Research Clinical Research Network. They evaluated the use of dexamethasone, 6 milligrams daily up to 10 days versus the standard of care. This graph on the right side demonstrates there's an overall decrease in mortality in the dexamethasone group. Based on the oxygen requirement at time of randomization, those who were on mechanical ventilation and oxygen experienced benefit from dexamethasone use. And this benefit was not replicated in patients not requiring oxygen supplementation. So based on the results of this recovery trial, the NIH and the Infectious Disease Society America guidelines have altered their recommendations and favored the use of steroids in COVID patients requiring oxygenation. One other treatment that has entered in the discussion is the convalescent plasma that has been used historically for other types of viral infection. So theoretically, antibodies to SARS-CoV-2 may help suppress the virus and may modify the inflammatory response. This JAMA paper was published early in May that demonstrated the potential benefit of convalescent plasma use in COVID-19. Post-infusion of the convalescent plasma demonstrated that there was normalization of temperature within 3 days in 4 out of 5 patients. ARDS resolved at 12 days post-transfusion, and ventilator was weaned within 14 days in 3 out of 5 patients. Now, there is another randomized controlled study in China that was conducted much later in the pandemic and was not able to demonstrate any significant difference between those who received convalescent plasma versus control arm. The 28-day mortality and time to discharge was not different. Actually, the clinical improvement within 28 days was not any different between convalescent plasma and control. The 28-day mortality and time to discharge had trends that favored convalescent plasma. However, it did not meet statistical significance. When they did a sub-analysis of the severe disease group, they found that there was shorter time to clinical improvement within 28 days and higher rates of clinical improvement at 28 days amongst those who received the convalescent plasma. Note that this study was terminated early due to the lack of new cases, and the authors concluded that the study may have been underpowered to detect a clinically important difference. In the United States, it has been used under the FDA Emergency Use Authorization. And per this paper published from Mayo, there was a low rate of serious adverse events among those who received convalescent plasma. The side effects included some transfusion reaction. Notably, the 7-day mortality rate was 14.9%. Clinical trials are still in progress, and we are still waiting for the results. So in summary, there's no approved drug yet for COVID-19. To date, clinical trials have supported the use of remdesivir as the antiviral therapy of choice and dexamethasone as the immunomodulator. Convalescent plasma may have a potential role in the management of severe COVID-19 pneumonia, and therapeutic options for mild disease in the outpatient setting remains lacking. And I want to acknowledge my colleagues at Yale School of Medicine who worked tirelessly during this pandemic, and thank you very much.
Thank you very much, Maricar. And thank you to all our last speakers. That was really excellent talks. So we have some time for like question and answer, and I think we have a little bit more time than we thought we had. So please just keep writing your questions on the side, and I think Edsel has been answering some of them. So let me just go over some that I think weren't answered. So there's one question. What is the main mode of transmission of COVID-19 in the Philippines and the community? And I don't know how you might interpret that question. And I guess that's for Edsel. Do you want to take that?
Yeah, I mean, I think that the evidence shows that it's still respiratory droplet more than anything. The aerosol data is suggestive. We know it happens in the hospitals, especially when we're doing aerosol generating procedures. But again, it can happen in areas with poor ventilation. If the G614 story does bear out that it is more infectious, 3 to 6 times more infectious, then maybe that we're going to see a lot more of this transmission. But most of the time it's still aerosol. Well, I mean, most of the time it's still droplet. What's concerning to me, though, is that we are seeing a lot more asymptomatic transmissions. We had a breakout in our laboratory, and the only reason we found it was because our research associate was going to the province, and they required a rapid test, which we don't really like doing because it's not very predictive, but it was IgM positive. So we went ahead and swabbed him, and it was positive. And we swabbed our whole lab. We had 5 people who were positive, none of whom had symptoms. So it's really strange. I think we're still learning more and more about this virus, and it's really hard to say what works and what doesn't, on top of the fact that the virus continues to evolve.
Okay, thank you for that. So actually, it dovetails into that one question, which I think all the speakers can feel free to answer. Which test would you deploy to open the economy? And I think there's several ways of thinking about that.
Yeah, I mean, I think it's a balancing act. I think, you know, you have to balance the costs versus how accurate it is. I know, I think Maricar would know, and Vince, our undergraduates, they just have testing, right, recently. Surprisingly, they're going to test them, you know, several times a week. I suspect that's going to be PCR testing to see whether you pick the positivity. That's a lot of, you know, resource put into having schools, kids going back, you know, it's, so there must be a better efficient way to do it. And so I've already heard a lot of comments about that decision. And so the antibody test is ideally would be good, but I think it hasn't been shown to be highly reliable yet, and to see whether there's any reinfection despite having antibodies. So I think those studies still need to be done, right? And so so I think it's really having careful social distancing, mask use, you know, being careful.
Any thoughts?
Yeah, maybe. Yeah, maybe I'm just going to add. I mean, very early on, a lot of us were kind of aghast that WHO said test, because part of the problem here is that our tests aren't perfect. They're 62% sensitive for an NPS on the day of symptoms. After 3 days, it's 80%, and then it's lower than that afterwards. So there's really no good way to pick up everyone. And if 40% of people are asymptomatic, what does that tell you? So someone's going to get through. And you know, we've been pouring so many resources into testing. And even if you get that test back in 24 hours, you could actually get infected the moment you walk out of the lab. So it doesn't make any sense that there's so much focus on testing when we should be putting resources into isolation, into contact tracing, into making sure that everyone is wearing their masks, and in technologies that can further prevent transmission. So I think it's all misguided. It's particularly bad for resource limited settings, because we've been pouring all our money into these molecular tests that aren't perfect and acting like it's we could test our way out of this pandemic. We can't. It's not going to happen. So the best thing to do is to put resources into taking care of the sickest people and make sure that they survive. We need to reinforce our healthcare systems, which are woefully already inadequate at normal times. And so we can't take care of COVID patients. We can't take care of non-COVID patients. That's where the deaths are going to come from. It's not all these 80% mild people that are going to get better on their own. So I don't think that you should even try, or it's realistic to test everyone that you think should be tested. I think we should really prioritize our tests and put the tests back in the hands of the doctors rather than on the policymakers who are just deploying it without knowing what it does, and missing a lot of false negative cases and generating a lot of false positives. So that's my soapbox. Sorry about that.
No, that definitely makes sense. So I think we'll go to the next question, which I think also will generate a lot of answers from you guys. So for all 3 speakers, could you describe how a surveillance program should work? Like how often should you test if you're actually surveying? Where it's that whole concept of, is it that every 2 weeks? Do you do it when people are just symptomatic? What are your thoughts on that?
I mean, I think it's still the same thing, right? I mean, it's if you want to know, like in the community, how prevalent is the virus, or like do you have a mutation, for example? You kind of have a solution to what to do with that information, right? And so I know I mentioned about the sewage testing just to get a sense of what the community has and when will be another rise, but even if knowing that information, you don't have a good sort of public health prevention, it's not going to help anybody, right? So, and and so I think it's still unclear. I don't know. Other folks have any other additional thoughts?
Yeah, again, the issue really is, again, when you walk out of the lab, even if your test is negative, you can get infected right away. So there's no way you can capture all these. I know some hospitals are testing every 3 days. The NBA is testing every day. That's a little bit much. But again, who needs a test? If they're healthy and they're young, the issue really is you're not testing to treat them, because if it's mild then it's not a big deal. You're testing so that they don't spread it. So if you prevent the spread with masking, social distancing, and everything, then you mitigate the problem of having to test all these mild people. And even if you test all these mild people, there's going to be 40% or more walking out there who are going to test false negative anyway.
Okay. So with that question, Edsel, the question is like, any seroprevalence study being done in Manila right now?
Yes, actually, former Secretary of Health, Manolet David, is doing seroprevalence. They initially were using rapid tests, which we know some of them turn negative after a few weeks because of lower titers. And I think they're trying to supplement that with ELISAs. I haven't seen the final data yet, and I don't know how they sampled. Anecdotally, the city of Manila started doing CLIA testing on people, and they tested about 4,000. And of course, these were self-selected; it's not a random sample, and about 600 were positive. So yeah, that's a fairly good number. So we'll see. But again, I don't know how that's sampled. I mean, those could cluster in households as well if you have full households getting tested.
Okay. And then maybe Maricar, I think this might be for you. So what role would monoclonal antibodies play in treating COVID-19?
Yeah, so actually this morning we found out that kind of disappointing result from this tocilizumab clinical trial. I guess, my colleagues and I are a little bit biased because we think that, based on our local experience that, and based on our local data, that we think that it may have helped. I think, the use of this immunomodulator really depends on when you actually initiate it. When you initiate it very much late in patients who are already critically ill and mechanical ventilation, it's almost like the cytokine release storm has already hit like so hard that there's no way of almost like reversing it. So I think our approach of what we have done when we have given it early, we believe that it actually helped in preventing patients getting intubated. So, but I think in the total scheme of things, in terms of like treatment approach, I think it's really a multimodal approach. It's really a combination of antiviral, if given early, and kind of giving this immunomodulator at the right time. And sometimes, like, the right time, when is that? It's kind of pretty golden. Mm-hmm. So one of the actually our experience has been using CRP as a good predictor. It's almost like you can see the pattern of the CRP going up and the O2 requirement going up, and then you kind of hit them with with tocilizumab. But currently our approach has changed because we have to follow what the data tells us, which is actually now we shifted to the use of steroids. So we are actually in the process of evaluating, our experience too, based on the combined approach of the remdesivir and the tocilizumab, and also look into our data because initially we have used also steroids in a reverse fashion, wherein we hit toci first and dexamethasone after, or any glucocorticoids afterwards. So those are, it's not a randomized control study, which is the ideal setting, but during the early days of the pandemic, obviously, we didn't have the infrastructure to carry out any of these studies.
Yeah, I second the motion. I love tocilizumab. I mean, it's worked really well for me as well. But aside from tocilizumab, which is an IL-6 monoclonal antibody, the one that I'm really excited about are the direct monoclonal antibodies against the virus. Monoclonal antibodies against the virus. That's what I was
thinking.
And I think also they're thinking about combining them. And the one that we have the most experience with, well, not personal experience, is HIV. And we know that the broadly neutralizing antibodies in combination do work. So there is some proof of concept in that sense. And the nice thing about these antiviral monoclonal antibodies is that they're going to have long half-lives. And so they're kind of, you could use them as a bridge, as like passive immunization, particularly for our healthcare workers. If they do work, then let's give them to our frontliners, because then, you know, they'll be protected maybe for a month or 3 months at least until the vaccine comes out. And it's almost a bridge to the vaccine in that sense.
Okay, so I'll probably keep it to 1 or 2 last questions, but there's one where any comments about the herd immunity that seems to be happening in Mumbai? How was our seroprevalence testing going on the country sponsor? Oh, well, that was the same. Okay, but what do you guys think about the herd immunity in Mumbai? Is that possible actually?
I think we know too little about it. We also don't know if these are neutralizing antibodies or non-neutralizing antibodies, and how long they last. And the more important, as has already been put in Nature and all the other papers, is T-cell immunity, because that's what really has an antiviral effect.
Okay, and then the last one, this will be an easy one. And this will be more of a, like, what's your opinion? So which country is doing the best job of isolating and minimizing the impact of COVID-19? I say it's America, actually. So you guys can take your pick of the other ones left.
Yeah, I think it's too early to tell, because I mean, even the best places like Taiwan and so Taiwan, South Korea, Japan, Australia have had secondary outbreaks. Hong Kong is in trouble right now, and we did okay initially. And the initial, you know, you can see if you go back to the by date of onset of our cases in the Philippines, the ECQ really did bring the cases down. Unfortunately, now that we open up, and then maybe the increased, so it's very difficult because each country is unique. It has its own dynamics. You know, like Manila, Mumbai, those are people in very close settings. I mean, you look at Singapore, they had a good response initially, and then they had an outbreak among the immigrants who were in close quarters. And, you know, it only takes one, and this virus is an infectious disease. So I'm not going to write any report cards at this point because we are far from over.
Yeah, I mean, I agree, because I think in the Northeast, we initially had the big spike, right? And now we're on the bottom, which is hopefully will keep, but we're not like going to celebrate yet because we're worried about another increase. And some people say it might come in August, as you know, some of the students come back, and we have hotspots in other parts of the country. And so this is not, I agree, this is not over until I think we have a good vaccine and people follow the preventive rules. So.
Okay, so I think at this point, a few questions still came up. What we can do is like maybe the speakers can just look at it and then just try and answer it like written down. And I'll give the floor to Vince again for the next session.
Maima, thanks for moderating the Q&A. A question came up. I'm really interested what our folks think about this. Success with convalescent plasma. Maricar, I'll put you on the spot.
Yeah, so I think it's too early to say. I think just based on our local experience, it's hard to tell because these patients have already been hit hard with so many agents, and then they will get the convalescent plasma on the very latter stage. So we don't know exactly if the convalescent plasma really did help, or maybe this is just basically the patient's immune system recovering on its own. So I think our local experience was kind of equivocal, I would say. So I think we're still waiting for that clinical trial to really, show whether it's beneficial or not.
Yeah, I agree.
Experience. Yeah, go ahead.
Yeah, it's kind of hard to say also because each patient is unique. Their titers are going to be different. We don't know what proportion of it has neutralizing antibodies, and on top of that, these are really hard to control because you're not going to give a sham convalescent plasma to anyone. It's not going to be a nice randomized trial. So yeah, it's really tough.
Okay, so thanks everyone. Thanks for our first three speakers. So we have to move things a little bit. Our next speaker isn't around yet. He's a pulmonologist, and I don't know whether he got stuck with, hopefully not with a COVID patient. But we're gonna skip him for now. We're gonna move on to our 9:20 talk, which is almost just in time. So our next speaker is Dr. Pipo Bundoc. His background is sort of interesting. So he's an orthopedic surgeon, but now he's going to talk about ventilator. So, Sir Pipo, floor is yours.
Good morning. Can you hear me?
Yep, can hear you well.
Okay. Good morning. So it's so nice to see most of my students excelling in their fields, and experts in everything. I would like to thank the Philippine-American Academy of Science and Engineering for this opportunity to share our humble stride in facing the COVID pandemic to provide Filipinos some fighting chance in these extraordinary times. Allow me, however, to apologize that I will be talking in generality to conform to some of the provisions and restrictions related to our registration with the Department of Science and Technology. The standard of quality of treatment when patients develop difficulty breathing is to provide high-flow oxygen through a nasal cannula, observe the patient, and provide all medical therapeutics that will open the airways. If this fails, the next thing one will probably see is the initial utilization of an artificial manual breathing unit known to many as the Ambu bag. A study done in my university hospital some years back of 320 medical interns who are required to undertake mandatory basic life support training revealed that only 1 out of 12 can correctly deliver the correct volume, pressure, and rhythm in manually ambu-bagging simulation dummies. And for those who eventually got it correctly, it took them only 10 minutes before they started faltering, either due to poor attention or fatigue. Simply speaking, manual ambu-bagging is not an easy task. One needs to multitask in minding the volume, pressure, rhythm, and even the position to perform the procedure. At best, one can only perform it properly for not more than 10 to 15 minutes. Now, this becomes ultra dangerous when we relegate the task to a patient's relative, because even if we medical practitioners cannot do it properly and consistently, how then do we expect a wary and a distraught parent, spouse, a grandparent, sibling, or even a friend to do it safely for their sick relative? Now comes the big question. Just how many patients do we think have died not because of the disease, but because of proper ambu-bagging? In a nutshell, I have given the first part of my talk, our motivation, followed by our design, and lastly, our progress. The University of the Philippines COVID pandemic response team predicted in early April that if we will not be able to flatten the curve, roughly 13,000 patients will need ICU treatment. And roughly half of them, 6,000, will be needing ventilatory support. Unfortunately, for a population of 110 million, the country only has 1,517 available respirators in the country. But it is encouraging to see that 2 locally designed ventilators, dedicated, however, for prolonged critical respiratory care, are now undergoing clinical tests in my university hospital. Yet, we have to realize that most of patients who will be hooked to ICU dedicated ventilators will first have to be probably passing through a stage when they might have to be ambu-bagged. For that matter, manual ambu-bagging, with all its inherent faults, will still be a de facto procedure as an initial respiratory support for most patients, while they are in transit from community to the hospital, while waiting in the emergency rooms for admission, while the respirator is still not available or being prepared, or when the patient has to be brought out of the ICU for ancillary procedures like X-rays and CT scans, or when the respirator malfunctions or power fails. And most obviously, with our current pandemic, the danger of infecting a relative who is ambu-bagging the patient is almost a certainty. Thus, we believe that all of these initiatives all over the world that use an ambu bag as a volume generator has its undervalued merit. The Vents for Philippines collaboration strongly believe that we have to come up with a device between manual ambu bagging and the ICU ventilator that is interim, transitional, or provisional, as you may call it. A device we generically call a mechanical ambu bag insufflator, MABI for short, a device that will simply supersede manual ambu bagging, that will deliver consistent volume, rate, and pressure of air, and a device safe for the patient and his caregivers. Now, cognizant of the fact that patient safety should be paramount in our mind, we have to turn to foreign guidelines of what such devices should constitute, because none is locally available. Earlier in March, we only had this UK guideline from the Department of Health and Social Care for rapidly manufactured ventilatory support system. We thought, cool, we can easily achieve all of these parameters. But late April, the Medicines and Healthcare Products Regulatory Agency of the same country gave a more stringent specification for rapidly manufactured ventilator system. But these refer to ICU respirators for long-term treatment, not the interim device that we are proposing. We also took notice of your bulletin number 14 regarding the validation and approval of low-cost ventilators. And of course, the statement of concern given by the Philippine College of Chest Physicians, the very group of people whom we might expect to utilize what we are hoping to achieve and produce. And that was how this collaboration of individuals, private groups, engineers, doctors, and the academe came about to become the Vents for the Philippines collaboration. When we started our endeavor, our biggest challenge is the lockdown of all possible sources of materials. All the materials we used for prototyping were off-the-shelf surfaces. Nevertheless, we finally huddled to our concept design. A, our device is going to be ambu bag based, followed by B, the mechanical component that will compress the bag, which is controlled by C, the digital backbone that runs B, and finally, D, the encasement that will incorporate A, B, and C as one device. Let us go back to A. Not all ambu bags are created equal. We will require one that has at least a pop-off safety valve that releases at 40 centimeters water, or preferably a bag that has all three, including a PIP valve and an inspiratory gauge. Next is B, our mechanical unit. We have a choice as to whether it will be hydraulic, pneumatic, or mechanical. We chose the simplest, a mechanical system run by a motor. We sat down and deliberated what would be the motor and the transmission system. Choosing the motor was not very hard for us because we had several working prototypes ready using each one of these even before we collaborated. And we had experiences in its pros and cons. And we unanimously chose either a stepper or a servo motor. Our choice of mechanical transmission system relied on the choice of simplicity. We opted for a single pincer arm, which is connected directly to the spline and axle of our motor, so that the manufacturing will not entail machining and milling of gears anymore, making it a lot cheaper. Then C, the electronic and digital backbone of the device. Our engineers created a digital circuitry that will send the necessary signals to our mechanical unit to generate the necessary volume, the necessary respiratory rate, the right pressure, and integrate all of this to sensors to form into a graphic readout and an appropriate alarm system when it malfunctions. All this could not have been done without the hospitality of the Technological Institute of the Philippines, where almost all bench work was done in the Rapid Prototyping Center at their Technicore facility. And of course, I have to say, the dedication of their faculty engineers who performed dozens of iterations to synchronize the digital and mechanical output of our prototypes. And finally, our encasement, which we want to be portable and easily handheld. One that you can bring anywhere, place on top of a bed, bring in an ambulance, place in a desk beside the patient, simply one with a very minimal footprint or none at all. We have started from scratch and since then has arrived with several prototypes. Shown here is version 4 and 5 of our prototypes. We are now in version 6. Probably we will be submitting version 7 to DOST for bench testing sooner. All in all, we want to create is a device that can universally incorporate most kind of ambu bags. A very simple design that is easy and economical to manufacture, yet maintaining high function that is safe for our patients, and one that can be brought out into the field even in times of calamities and disasters. Some functional features of our device will be dependent on an ambu bag, specifically the pop-off and the PIP valves. Our device does not compete in any way with the other 2 endeavors that are now being tested in our university hospital. It is, in fact, meant to be an auxiliary support device to these 2 ICU ventilators designed for prolonged critical respiratory care, which we hopefully see manufactured soon for our country. One we can say proudly Philippine-made. As we have reiterated several times, since our proposed device will not be used for long-term critical care, we wish that the Department of Science and Technology and eventually the Philippine FDA will see the wisdom why we do not have to incorporate certain features in our device. For one, we do not need to have a CPAP and a BiPAP mode, which are necessary in weaning patients from prolonged ventilator support. Ours is not going to be used for weaning patients from ventilator support. We also can dispense of heaters and humidifiers, which is not critical in the short-term utilization for device. And we do not intend to have a trolley footprint for something we wish to be portable. This is our long road in realizing our goals from institutional registrations down to the FDA approval. So far, we are just about to submit our prototypes for bench testing. More so that we hope that the Department of Science and Technology will agree with the issues that I have raised so far in considering our project as a totally different concept. A much more simple device, thus a totally different device compared to ICU ventilators. I would like to end my talk by going back to this slide, which I have shown previously. For heaven knows how long, this picture has been an all too common sight in our healthcare system, such that we believe that it is normal. No, it is never going to be normal. Nobody has ever done a health technology assessment, shall we say, in this case, of an elderly husband ambu bagging his wife. I strongly believe that the measure against which an innovative product is to be gauged against is not the standards of Medtronics, of Philips, of Siemens, or Draeger, but the heartbreaking patience of a sorrowful mother ambu bagging a daughter, or the filial affection of a son snatching his father from the claws of death. Our fellow Filipinos deserve something better than manual ambu bagging. Allow us, help us to provide our countrymen with something better. Thank you very much.
People, thank you very much for that wonderful talk. Like, you're giving me like memories of my days in PGH where we used to ambu bag these patients. It's nice to see that innovations are being made to make lives easier for our patients. And you know, as a pediatric intensivist, like ventilators are lifelines. So I know where you're coming from. So moving on. So people mentioned the OstreaVent. So that's going to be our next talk. Bob Dizon will give us an overview of the OstreaVent from its origins to where it is now. So Bob Dizon is the Executive Director of the Metals Industry Research and Development Center at the Philippine Department of Science and Technology. Bob.
Good morning, everyone. Can you hear me, Dr. Vince?
Yep, I can hear you well.
Yes, thank you, Dr. Vince, for inviting me to participate in this year's PAASE conference. It's an honor to be talking in front of distinguished scientists, engineers, and medical professionals. I'm here to tell the story of how we developed the OstreaVent 2, the one you see on the right, from the original OstreaVent infant ventilator that is on the left. So Dr. Bundoc and I are using the same picture. Anyway, way back in 2012, Dr. Enrique Austria, a professor of pediatrics at Wayne State University and a neonatologist at Hasso Women's Hospital and Children's Hospital of Michigan, formed a team of mechanical and electrical engineers from the University of the Philippines to develop the OstreaVent infant ventilator. I was fortunate to be a part of that team. By 2015, Dr. Austria, through the Breath of Life Foundation that he founded, started distributing the OstreaVent to different hospitals in Metro Manila. At present, more than 70, that's 70 units have been distributed and are being used in various hospitals all over the Philippines. Unlike commercial ventilators wherein the device is placed between the supply of air and oxygen mixture and the patient, the OstreaVent is placed after the patient. Instead of directly controlling the delivery of the mixture to the patient, the OstreaVent controls the exhaust air out of the system. Let me show you how this is done. Okay, so the OstreaVent is protected by a utility model registration, so I am at liberty to disclose its operation. The main component is the solenoid valve. It is used to alternate the flow of exhaust air mixture between a high pressure path and a low pressure path. With the continuous input flow of regulated air and oxygen mixture, when the solenoid valve is closed, and because the flow to the lower path is restricted by the small opening of the PIP valve, most of the mixture is forced to flow into the patient's lungs here. The PIP valve is a throttling valve, and it controls the PIP by adjusting this small opening. When the solenoid valve opens, the airflow tends to move toward the lower pressure path, that is through the PEP valve. The air inside the patient's lungs also tends to be expelled out. The PEP valve is also a throttling valve, and it controls the PEP by adjusting its opening. The solenoid valve is controlled by a programmable logic controller, or a PLC, which can be set to determine the number of breaths per minute and the length of inspiratory time, or I time. This PLC also takes the real-time pressure in the system and compares the value to user settable limits and issues over and under pressure alarms accordingly. These are the features of the infant ventilator. It is a pressure limit time cycled ventilator. It can do 0 to 80 breaths per minute. The PEP can be set from 0 to 15 cm of water. There are audible and visual alarms with adjustable alarm limits. It has a bar graph display of real-time pressure along with the PIP and PEP alarm limits. It has a colored 7-inch touchscreen user interface. Excuse me. It can display the pressure waveform in real time. It records internally up to 3,000 data logs, which can be retrieved through a USB flash drive. The OstreaVent, however, has limitations which make it unsuitable for use for COVID-19 patients. First, it was designed for infant use only, while most of the patients are adults. It is pressure controlled, but the requirement is for volume controlled ventilators. It can handle only up to 10 liters per minute airflow rates, but adult requirements can go as high as 70 liters per minute. There is no tidal volume measurement. There is no bacterial or viral filter. Inspiratory time is up to 0.8 seconds only. In the latter part of March of this year, during the start of the community quarantine, Dr. Austria formed a new team to develop an adult version of the OstreaVent, which can be used in this pandemic. I was called again to handle the electrical engineering aspect. That was the time I met Dr. Fausto, our host in this session, who became part of the medical team. Anyway, the new OstreaVent has to meet the following requirements. It should be for adult use also. It must be able to handle airflows up to 70 liters per minute. The maximum inspiratory time of 0.8 seconds should be increased to 2 seconds. There should be an option for volume controlled operation. Of course, with volume control, there should be tidal volume measurement. There should be bacterial or viral filter. Now let us go back to the operation of the infant ventilator. Increasing the maximum inspiratory time from 0.8 to 2 seconds is quite simple. As mentioned earlier, the operation of the solenoid valve is controlled by the PLC. Thus, changing the maximum I time was just a matter of changing a few codes in the PLC's program. On increasing the flow rate, we did not have any idea on what the effect of the increased flow rate on the ventilator will be. Thus, we subjected the ventilator with around 30 liters per minute of flow to see what would happen. We noticed that the pipes, the tubings, the components, the valves, all can handle the higher flow rate. But we also noticed that it takes a while for the pressure to drop from PIP to PEP upon the opening of the solenoid valve. We therefore had to add a means to improve the PEP decay time. So you see here, there's the pressure takes a slow time to decay. So we had to add a means of increasing that decay time. At this point, I cannot disclose the specifics of how we did that, as we will be filing a patent for it. And nonetheless, we were able to meet the required PEP decay times with this contraption. As explained earlier, the OstreaVent comes after the patient and not before it. The use of a filter is therefore not as a precaution against the transfer of microorganisms from one patient to the next user, but against their release to the surrounding air. The question was where to put it, at the input or at the output. At the input, it will prevent contamination of ventilator components. The filter, however, may affect pressure and flow readings, especially when it becomes clogged. If at the output, there will be minimal effect on the ventilator performance. The drawback is that microorganisms, the virus, might accumulate inside and will be difficult to disinfect later. So we opted to put that at the input. We devised some workaround to minimize its effect on ventilator performance. Again, I cannot share with you how we did it this time. Volume measurement and volume controlled operation were the main challenges in the development of the OstreaVent 2. Again, I cannot disclose how exactly we did it due to intellectual property concerns. But let me just share that it was just a matter of choosing the appropriate sensor and feeding the data to the PLC, after which everything became a programming activity. So let me show you the home screens of the two OstreaVents. For OstreaVent 1 on the left, the user can set the BPM and the I time. The bar graph displays pressure in real time. The user can set the alarm levels for the maximum and minimum PIP and PEP to the 4 red lines. There are also displays for mean airway pressure and PIP and PEP. Take note that PIP and PEP are not set through this touchscreen, but through their corresponding throttling valves. There's also a button to go to the pressure waveform screen. For the OstreaVent 2 on the right, you can see the additional bar graph of the volume. There is also an option to select between high time or tidal volume modes, which are essentially the two operating modes, pressure control and volume control. There are still the map, PIP and PEP displays, but now there is the tidal volume. There are also additional buttons to display not just the pressure waveform, but also the volume and flow waveforms. We also added a shutdown button to avoid unintended powering down of the unit. So here is a glimpse of the two OstreaVent models in operation. You can see the home screens as well as the waveform screens. On the left, you can see the pressure waveform of the OstreaVent 1. On the right, you can also switch from pressure to volume waveforms. My engineering team, through constant guidance by the medical team, was able to fabricate the OstreaVent 2 prototype in just 1 month. That is through April of this year. By May, we were trying it out, clearing all the bugs, addressing the comments and suggestions of the medical team, and other requirements. We tried it twice on the Michigan lung simulator of Dr. Balgos in PGH. By June, we were ready to have it certified by the DOST accredited company. Sadly, up to this moment, we are still waiting for our schedule for the certification procedure. That's despite my organization is under the DOST. Apparently, there are so many things we need to comply with, even if they are not within the scope of our claims. Anyway, the remaining tasks after certification are clinical testing, which, by the way, we already got the approval to proceed from the UP Manila Ethical Review Board. We will also be producing 5 more pre-commercial units for field trials. So 4 of these will be used in the field, but 1 will be used by my team in developing a new model with assist control capability. So that's all for the development of the OstreaVent 2 ventilator. Thank you for listening.
Bob, thanks for that wonderful talk. So I was able to touch base with our other speaker. He's trying to log in, but in the meantime, I'm gonna turn you over to Maima first, and then let's start the Q&A. Once he's logged in, we will proceed with his talk and have some more Q&A after that one. Maima?
Yeah, okay. So we can take a few questions while waiting. So let me see. There's one for Dr. Bundoc. When do you think you can field test the device, and how does it compare to Ayala IMI UK ventilator device? I don't know if
the Ayala device is actually a BiPAP device, whereby they actually gave out all the blueprints and everything. So it's something that you don't have to start from scratch. And like compared to what we're doing, we really have to start from scratch. And when do we think we could have it field tested? No, like what Engineer Bob Dizon said, we are running into so many issues, and much to his frustration, where since he belongs to the same group who's going to validate ours, it's just a question of really complying with all of these issues. That's why even our talk has to be given in a very general tone, so that we will not be disclosing too many things that are being prohibited from us from really giving out to the group. So we're looking at something like another 4 weeks or 5 weeks before we can probably comply with all the things that the DOST is asking from us.
Okay. And so follow-up to that is, would this machine need calibration often to make sure it is delivering the right pressure? And then also with that whole, the bacterial viral filters, can it be decontaminated if this is used for COVID
it
patients?
should be those are things that are being asked of us now. Our engineers are looking at something like auto calibration every time you change the disposable Ambu bag, because the Ambu bag will be disposable. And of course, our filtration will contain the HMEF filtration devices, and those are issues that we really have to comply with for patient safety.
Okay. So I think in parallel, the question for Engineer Bob, it's how is the OstreaVent 2 sterilized against COVID-19 in between patients?
As I mentioned earlier, it doesn't really need sterilization because it comes after the patient. So the next patient will not really be exposed if there is contamination inside the ventilator. But nonetheless, we put in a filter, a replaceable filter. So maybe Dr. Vince can answer if that's really necessary to sterilize the unit.
So wait, can you repeat the question?
Do you need to sterilize the unit? No. So basically, do you need to sterilize the OstreaVent when it already has a filter? But I guess
right. So the way we sort of designed it is the unit itself should be free from contamination, right? So you need to change everything from the air supply up to the filter, because that's all disposable. But theoretically, beyond the filter, it should be clean, and you don't need to decontaminate that part.
Okay. And then, so I think this one is for the OstreaVent as well. Have you done proper risk assessment as part of the design control documentation?
Oh, what do you mean?
Proper risk assessment, yeah. Maybe this question is from Al Serafika. Maybe you can clarify what you mean if this question, risk assessment as part of the design control documentation. I don't know either.
Well, I don't exactly
understand the question.
What risk he is referring to, but
okay.
the design
of the ventilator. I can clarify.
Okay, there.
Bob, when we design medical devices, we do a design control methodology for documentation so that you'll have your validation and verification for your device prior to clinical trial. So that's a question that I had with your group, being mainly an engineering group at the MIRDC. If you've done, together with Vince, you can answer this as well. If you've done after the finishing the device design and the outputs are being measured, looks like you've got to the OstreaVent 2 with the right outputs already, and the tidal volumes, as you had earlier predicted when we were talking about this in March. Now that you have the device working, you're going through certification, which is actually validation for the medical devices. That's where I'm asking the group, have you done proper risk assessment? What if parts of the device fails? What would happen? What's the mitigation? What's the incidence of it happening so that you can do the proper mitigation? And that's why you're probably having a hard time or taking longer time to get the certification, because they have to go through all this documentation that you should have generated during the design phase of your device. So that's why I'm asking. So if you don't know what risk assessment is, that's probably you're gonna get asked of that prior to you guys getting into the clinical.
As I showed you in my presentation, before developing the unit, we listed down the objectives, the requirements. So those requirements came from the medical team. It's not from the engineering team. Those came from the medical team.
Oh yeah, that's your user need and your design input that came from the medical group. Correct.
Yeah.
Now, and then after during the development, we are in constant communication with the medical team, presenting to them, this is the objective, this is the problem, this is our solution. Is this fine? Is this good with you? So that's how we proceeded with the development.
So,
Yeah,
Bob, I can help you with
correct.
that one. So, also before all my involvement with this, like I really had no idea what this like risk mitigation is all about, right? So, let's, as a clinician, it's like give me a vent, I'll use it. That's where I was coming from. And then, you know, as I got exposed to a lot of this stuff, then all these like terminologies came about. So I got this from, I think it's the industrial engineering group, right? So that's where I got this like whole risk mitigation strategies. So we basically have that with the OstreaVent. It's just not termed that way. So part of the requirements for the OstreaVent actually includes all of those, but they just use different terminology. But I can tell you that the issues that we're encountering is not from that, you know, part of the equation.
Okay, good.
For
Thank
for
you.
for our reference, we're using PNS ISO 14971 2016 risk assessment reference form for the ventilator that we're trying to produce.
Thank you, Dr. Bundoc, on that one as well. Because that's where I wanted you guys to at least get a little bit of acceleration, because that's going to be the stumbling block for you guys to get an FDA approval and getting into the clinic. And I'm glad that UP Manila has already approved OstreaVent 2 for ERB, but I think they will require a similar certification. Unfortunately, here in the Philippines, we don't have that investigational device exemption that we normally get in the US for testing medical devices. So you guys are a little bit in a more difficult situation. That's why I'm wanting to help you eventually. But we'll see. Thank you very much for both of your updates. Good job,
Thank
guys.
you very much. Yeah, very difficult situation.
So we have Dr. Balgos online. I made him co-host. He's actually muted right now. I'm gonna unmute him. Sir Boons, are you ready to give your talk, or you need a couple more minutes to settle down?
Well, yeah, I can just share the screen.
Okay. Okay. So Maima, let's hold up on the Q&A for now so that we can hear Dr. Balgos. So, sir, people already mentioned this. So there are two ventilators, locally made ventilators. We've already heard about the OstreaVent. The other one is the Ginhawa vent, and we have Dr. Balgos who developed the Ginhawa vent. So Dr. Balgos, pulmonologist, he used to be one of my professors, just like Sir People. So Dr. Balgos, the floor is yours.
Okay, can you see the slide?
No, I can still see you.
How about now?
I can see you.
Okay, so while I'm working on it, I just want to briefly mention that the PGH pulmonary group has been doing research on and making projects related to the manufacture of ventilators as early as the 1980s. And
Yep, you can see your screen now. You can just put it in presentation mode. You're all set. There, perfect.
You see it now?
Yep, you're all
Okay.
perfect. Thank you.
So, well, many of you are doctors, but I just briefly want to show you that we felt that there was a felt need for ventilators early on because as much as 50% of ICU patients may need a ventilator, there was a big demand or need for ventilators even before the pandemic. So in 1987, we already made an Ambu bag resuscitator together with some engineers from the UP College of Engineering. And in 1993, we shifted because of the felt problems that we saw with the Ambu bag insufflator to a bag-in-a-box model, which was the basis for which we were able to get funding from the USD. And eventually in 2012, we were able to come up with this initial Ginhawa prototype, which was already a servo ventilator. It was, however, lacking in some of the features that we wanted because we wanted to put in better features so that it can really be a truly servo ICU ventilator. And so this is what we envisioned. At that time, we were looking at the one breath ventilator that is being developed by Stanford. And they were supposed to come up with it also as a low-cost ventilator. And so we initially just wanted the basic ventilator settings of CNP, assist control, SIND, CPAP, and P. But as we were pushing through with our development, we have actually improved. And this is now our newest prototype that is already going to undergo more detailed and focused bench testing. Soon we will have the ASL 5000 simulator from Ingram, from Ingmar, I mean, so that we can be able to simulate. So you can see here, we're using the test lung, Michigan Instruments. The problem with the Michigan Instruments test lung is it does not simulate spontaneous breathing. So the main advantage that we will have once our ASL 5000 simulator comes within the next 3 weeks is that we will be able to simulate normal spontaneous breathing in various clinical situations. So it can simulate high compliance problems, low compliance problems, high resistance problems. So even the situation of the two types of failure in COVID-19 respiratory failure, we can actually simulate. So the stage that we're in now is that we will be going through the field studies. We were hoping to start it this August, but because of the delays in the procurement of parts, as well as the ASL 5000, we'll probably be able to do more intensive bench testing by the end of September and start our field study by the first or second week of October. So basically what we're dealing with is affordable, safe and effective compact ventilator, which can be powered with both AC and DC power. We made sure that our battery will be something that could last for 8 hours, but we're working on a battery that maybe could be functional for at least 12 hours. The knobs are very easy to operate and can be used for children and adults because the tidal volume range is between 100 ml to 1,500 ml. Basically, what we envisioned before the pandemic was that the ventilator will be available for not only sale, but rental or lease to all options. And we have incorporated in the software self-diagnostic cloud-based data analytics so that our maintenance and troubleshooting team could follow up the function wherever in the Philippines it is located, for so long as there's an internet connection. And that will allow us to have full after-sales maintenance and also assure us of 24/7 parts replacement and repair services. So basically, this is our goal for the project in a nutshell. We were really targeting the government and small private hospitals who cannot afford the expensive ICU ventilators that we have now, while giving the full ICU ventilator capabilities. So we still have to look at continuing research and development and test testing. We actually already are looking at a model that will have graphics and touch screen to show us the pressure and other waves so that it will be easier for the ICU staff to monitor patients. This was a slide that I prepared around 7 years ago, and it was, looking at the need for ventilators for respiratory failure from pandemics, given at that time. But the use of a small, low-cost ventilator would be also useful as transport ventilators for home use of COPD patients, and maybe for those with obstructive sleep apnea. So this was basically our go-to-market strategy when we presented our project to the DOST as well as the AIN. And we are now at the stage where we are about to do field testing. So we're still on track, but we still have to do a bit more of surveys to the actual end users, which are the pulmonologists and the intensivists. And I think that's my last slide. Thank you for your kind attention.
Okay, thank you very much for that great talk. I think these last speakers were really interesting for me to see, where you see the ingenuity of the Filipino. So I think we can just cap up the session and finish up with some of the questions. Let me see. I think we left off in... So there was this one question where the question was, intubation seems to contribute to morbidity and mortality. Can the OstreaVent be used with just a face mask? And so I think whether Bob can just comment on that, whether it can be used with a face mask.
Yes, it was really designed for face mask use. I don't know if it can be used for intubation, but there has to be a tight seal of the gas, of the air. Otherwise, there will be leakage, and it will not generate the PIP and the PEP.
Yeah. Okay. And then I think the other part to that whole concept of intubation seems to contribute to morbidity and mortality. I think Edsel made that comment where, early intubation is discouraged for certain patients, but when people do need intubation, mortality isn't necessarily because of the intubation, but it's a marker of the gravity of the patient's condition. And then from Al again, he asks for Bob, the DOST, I don't know how you say that, Picard was planning to procure a ventilator simulation equipment. Okay. Was planning to procure a ventilator stimulation equipment from California, US in the discussions in April. Did it happen? And can Dr. Balgos and Dr. Bundoc, can they use that?
Actually, this our team, which is DOST funded, that ordered for this, that's what I call the ASL 5000 simulator. It's a simulator that is being used by major ventilator manufacturers worldwide. We wanted to order, and we did have initial funds from the DOST for 3 units, but because of the severe need for it. Our order was backtracked, and the first unit that we will have will be delivered only in the early part of September. That is what I was talking about in my presentation. This simulator is able to simulate a patient on a ventilator with pneumonia, with congested heart failure and alveolar flooding. So it can simulate many clinical scenarios without hooking the ventilator to an actual patient yet. And this is going to be really useful for the ventilator developers because it will make us more confident about our ventilator capabilities even before doing the field testing.
Okay. So at this point, does anybody have any other questions? I mean, if you do, you can just literally turn off, unmute yourself and ask for our last three speakers, for any of the earlier speakers. It's your last chance.
Jemima, before we entertain some more questions, I just want to ask Dr. Balgos and Charles, what are your thoughts about intubation and mortality? I know we were pushing for early intubation before. Now we're sort of holding off. What are your thoughts?
Yeah, I mean, I think in the beginning people were worried about transmission, right? And so they wanted to be a more controlled intubation, and so they want to do it earlier to prevent the spread. I think that resulted in a lot of patients who have less severe disease being intubated, and I think it sounded like it was a bad idea. I think, you know, there's a lot of use of high flow nasal cannula now, and there's a lot of use of non-ventilated patients doing proning prior to being intubated. So I think there's different modalities that we could prevent patients from getting intubated. And we've learned how to get more PPEs. And so I think that has helped to have it a little bit more wait until the patient need it. You know, some of our experience in the ICU, encouraging patients to do self-proning while on high flow on the awake patient, sometimes it's not perfect because they're awake and it's not the most comfortable and you don't do it perfectly. And so you kind of fool yourself sometimes because you know their numbers are good, but they're not really, and then people go up on the oxygen level, and but people underestimate how bad their lungs are, right? Because if they were intubated on a mechanical ventilator, you would know the numbers are really horrible with the PEEPs and the FiO2. So I think right now, I think people have a better idea of it, and there's a lot of talks about the compliance and how there's different physiologies of the lung. We also know that there's a lot of vascular disease, right? Thrombotic disease, and some of these patients having pretty good compliance despite really bad oxygen. And so I think the ventilator has really taught us a lot about the physiology. So without that, and people are on high flow, you don't get that physiology that much, right? You kind of underestimate the physiology.
So so Dr. Balgos is also a pulmonary physiologist. That's actually when I first met him. So since Charles started talking about physiology, Dr. Balgos, what do you think?
Actually, I'm not gonna talk about my experience, and I'll be talking about data from Gattinoni and co-workers based on the autopsy studies, I think from Italy and from the initial data from New York, which shows that they actually think that there are two phenotypes. There's the L and the H phenotype, where the L are the people who have less need for positive pressure and they just need high oxygenation. And these are the people who do not need intubation and who will just probably be benefited by just proning, even if they're not intubated. High flow nasal cannula, maybe even CPAP, and maybe this is where some of the insufflators will come in, non-invasive ventilation. But there's a subset that they classify as the H type who would need high pressures. So basically, the approach of intensivists and pulmonologists now is they have to look at the baseline physiology and clinical status of patients. So if there's, for example, a lot of interstitial infiltrates on X-ray and CT scan, and there is most likely going to be a lot of increase in resistance. These are people who must not be given high volumes and high peaks because they will develop barotrauma. So these are the people who will probably be given only 6 pills per kilogram body weight tidal volumes and peak that is no higher than 10 centimeters water because giving more will probably contribute to barotrauma, and this is where that generalized statement that ventilators are dangerous for these people, but actually it's far from the truth. There are really some patients who would need intubation and who need the standard ARDS protocols. But there are people, and I think we're now seeing more and more of these people who just need high oxygen and just some assistance for the breathing during the time that they are not yet developing the cytokine storm, and the lungs are still able to oxygenate.
Okay.
Back
Yeah, I
to
think
you,
that's
Maima.
I think that's interesting how in one of the questions where there's a surge now, so what now? But I think you can see how things have evolved over the last 5 months that we're in better shape to actually take care of the unfortunately likely second surge. So there we go. Now, I think that might be it. There's just one question about potentially fast tracking the approval and field testing process. I don't know if that's possible in the Philippines, but that has been raised. I don't know if anyone can take that question. So it's basically asking, with the current pandemic, can the Philippine regulating and approving agencies consider fast tracking the approval and field testing process? I don't think we have anybody from the approving agencies on this call.
I'm not familiar with all of the participants. Maybe some of them have worked with the FDA and have an idea whether this is possible. Just send me a message. I'll unmute you.
Yeah.
If anyone knows anything about the FDA in the Philippines.
Or raise a hand. Oh, Alter Africa raised a hand.
Yeah.
Perfect.
If I may, for our Ginhawa project, we have actually already requested FDA approval for our earlier prototype, and that was actually around 3 years ago. And we were given interim approval pending the human field studies. However, we were the ones who stopped the process because we felt that we had to give more features and safety features to our prototype. So we stopped. We asked for a change in our budget and project study, and therefore we are now working on this new prototype. So right now, the only advantage probably that this pandemic has given to us is that the DOST has assured us that although our initial target for our field study is 60 patients, because of the use of the ASL 5000 simulator, we are actually being instructed that after the first 10 patients, we are going to have a safety committee review the performance of the ventilator even in the first 10 patients and see if we can actually do early termination and already proceed to manufacture and mass production. And so I think the FDA, although I have not talked directly to them, we were talking to the DOST, will most likely give an early approval if we put together the safety committee, which is going to be composed of the FDA, the DOST, independent biomedical engineers and clinicians to evaluate the performance of the ventilator on the first 10 patients that we have.
So Al,
Thanks, Bunds. Yeah, I'm okay, Vince.
Yeah, go ahead.
Yeah, thanks. Thanks once for the update. Definitely, I'm hoping that we can get your Ginhawa already in the clinic if the need arises. I'm so happy to see 3 active models right now between OstreaVent 2 and the TIP model with Dr. Bundoc. And the fourth one, that's of course not discussed on the table, which is a manufacturing scale ventilator of Ayala IMI that was part of the UK Ventilator Challenge, was made available to the Philippines actually as early as June. They told me if we can find a clinical team to evaluate it. So we did ask DOH, so they can go ahead with their validation study. That way it can be in the clinic if the need arises as well. So with all these 4 options, I think I told you guys when we met on this one as early as in March, that this is a rat race. I mean, it's a horse race. Whoever gets in there gets used. And that's actually what the FDA has kind of intimated to us, that being that it's an emergency use authorization, similar to what the US is doing in diagnostic kits, in doing it in cure, as you've heard it from Dr. Malinis. And definitely the same applies, hopefully, on ventilators. Luckily, we're not needing it. We're only at 28%, I think, in utilization right now, and our doctors are doing a good job preventing them from getting to the point where they need intubation and ventilators. So I think, but nevertheless, the next stage would be clearly the regulatory and certification and validation and clinical study. And again, as I told you early on, I had a medical device company in the US for 20 years. That's what I do for a living for about 20. So I'm wanting to help you guys, not only from the initial design control documentation to the certification, and then hopefully get to the clinical approval for testing. And so good luck with all 3 presenters. And again, looking forward to assisting you guys in the near future. But thank you for actually joining us in PAASE. I'm past PAASE president in 2017, and I'm so happy that Vince came up with this roster. Good job, Vince. Thank you.
Thank you.
Okay, I think just one last question, and it's been a long night and morning for the people in the Philippines. So any data on acrylic box or barriers? I guess they mean for intubation.
Yeah, for
Vince, maybe you
intubation.
can just take this.
Well, I don't know any data. so
We don't
Efren
use
is
it
my
here.
classmate. So Efren is my classmate. I asked him to ask a question. Now it's fired back. Now I need to answer his question. I think there's I think in New England Journal of Medicine they had like some video intubating with an acrylic box, and it's a physical barrier, but I don't think I've seen any data to actually quantify how much comes out of that barrier. I don't know whether Dr. Balgos or Maricar or Charles have more information about acrylic boxes during intubation.
Yeah, sorry, I don't.
We don't use it here.
Yep.
I mean, they use it
Sorry
in the
about
ER,
that. Yeah.
but I don't know if there's concrete data of
Yeah.
showing it reduces the risk of transmission. I don't know if Charles. I'm putting Charles on the spot.
I've seen it used in our hospital, but I don't think they've done a study yet. I mean, I think it's not consistent in terms of the use. I mean, I think last time I was in the ICU, I saw them sitting outside the ICU. I guess I think the the concern of people are having full PPEs when they go in. The anesthesiologists who do a lot of the ICU intubations, they have even the full head cappers for it, and so they limit the number of people in the room. So potentially, but I think I haven't seen the data unfortunately.
Dr. Balgos was raising his hand earlier.
Yeah, I don't think there are studies on whether these aerosol boxes has prevented spread of the infection, but I would dare to say that because of the aerosol generation that is being generated during intubation as well as other procedures, I think the healthcare workers would be better off using what we call PAPRs, purified, powered PAPR, purified airway, air pressure respirators, where you know air is pumped in, which is actually through HEPA filters, and it creates a positive pressure in the face or in the shield, so that the aerosol is not the chance that the aerosol virus carrying the aerosol carrying the virus would be less likely going in. So I work and practice in a small private hospital, but we actually use the APRs. Although we actually have the aerosol boxes, but we still encourage or actually almost force our healthcare workers to use the APRs.
Okay, so it looks like we don't have any more questions. So one last chance to put in any more questions. 3, 2, 1, done. Okay, so that's it. So thank you very much for presenters for sharing their wisdom, and to all our participants. It's been a long night, but very fruitful. Well, day in the Philippines. Very fruitful, but very productive. So again, thank you everyone, and good night, and enjoy the rest of your day. Thanks.
Thank you very much. Thank you, PAASE. Good night.
Thank you too. Thank you.
Full screen. Is it full screen? Yeah. This is just